The line hypothesis states that at a random point during a somatic cell's development, an X chromosome is inactivated. Dosage compensation ensures that females will not produce more of the X-linked gene product than males. These X chromosomes are called bar bodies. Females have one bar body in each cell and males do not have any. This is Scruffy. She's a Maine Coon mix with a tortoiseshell phenotype. When we say mosaic expression, tortoiseshell cats are a great example of that. They're heterozygous at the X-linked trait, and random inactivation of either the black hair or yellow hair X chromosome results in that patchy tortoiseshell coat. Adding on a second trait, long hair is autosomal dominant and short hair is autosomal recessive. We're going to cross this female and this male and figure out the expected phenotypes of the progeny. Using Punnett squares, we can separate the traits and see the expected phenotypical ratio for each trait. Using the ratios we got from our Punnett squares, we can now do our branch diagrams. Our results are: a quarter will be tortoiseshell long-haired females, a quarter will be tortoiseshell short-haired females, a quarter would be yellow long-haired males, and a quarter would be yellow short-haired males. Two female progeny from this cross, as we can see, will both be tortoiseshell. However, they will not have completely identical coat color patterns. This is because X inactivation is random, therefore it's not possible for all their cells to have the same inactivation pattern of X chromosomes.